A fish, declared extinct for 66 million years, surfaced alive in a fishing net, not in a fossil bed, in a net. The 22nd of December 1938, the mouth of the Chalumnae River, South Africa. A trawler called the Nareen had just dragged its catch onto the dock at East London. Salt air, fish, smell, the ordinary noise of a working port. Marjorie Courtney Latimer was 24 years old, curator of the East London Museum. She came to the docks regularly, looking for specimens worth keeping. The trawler captain, Hendrik Goosen, had saved something unusual for her. It was buried under a pile of sharks and rays. She pushed through the catch and found it, 5 feet long, steel blue with pale white flecks, scales like armored plates, and 4 fins that were not fins, not exactly. They were lobed, fleshy, muscular at the base, more like limbs folded against a body. In her own words written decades later, "I picked away the layers of slime to reveal the most beautiful fish I had ever seen." She had no idea what it was. Nothing in her reference books matched it. She sketched it by hand, packed it in ice, and sent a letter to the one man she trusted, ichthyologist J.L.B. Smith at Rhodes University. Smith received her sketch on the 3rd of January 1939. He later wrote that he stared at it and felt the floor move under him. He recognized the creature immediately. It was a coelacanth, a fish the fossil record said had vanished before the first dinosaur walked the Earth. Smith confirmed the identification on the 16th of February 1939 and named the species Latimeria chalumnae after Courtney Latimer herself. The survival was astonishing. But 75 years later, a team at the University of Washington sequenced the coelacanth's complete genome and found something inside the DNA that made the fish's survival look like the smaller story. There is a specific gene sequence identified in a 2013 paper in Nature buried inside this animal right now. What it codes for and why it has never activated is what this record is here to explain. Here is the comfortable explanation science settled on. The coelacanth survived because nothing needed to change. It found a niche in cold, deep, dark water below 150 meters, and that niche never shifted. No predators, no competition, no pressure. Evolution, the argument goes, only moves when it must. Remove the pressure and the clock stops. The label that stuck was living fossil. Darwin himself coined it in 1859. The idea is elegant. Some organisms reach a design so efficient that natural selection simply loses interest. The coelacanth, in this framing, is a biological museum piece, alive but frozen. For 60 years, that explanation held. It was satisfying. It was tidy. Then, in September 1997, Mark Erdman was walking through a fish market in Manado Tua, Sulawesi, Indonesia. He was a marine biologist on his honeymoon. He saw a large, dark fish laid out on a market stall under bare overhead bulbs. Rough scales, lobed fins, that unmistakable silhouette. He photographed it. He sent the photograph to colleagues. Nobody could say it was the same population from the Comoros Islands, 10,000 kilometers away across the Indian Ocean. By the 30th of July 1998, a second Indonesian specimen had been captured alive. Lab analysis confirmed it. This was not Latimeria chalumnae, this was a separate species, Latimeria menadoensis. Distinct coloration, subtly different proportions, genetically diverged. A frozen relic does not cross an ocean. It does not speciate. Then came the 18th of April 2013. Chris Amamiya at the University of Washington and Jessica Alfeldi at the Broad Institute of MIT and Harvard published the complete coelacanth genome in Nature. The data showed something that broke the living fossil model entirely. The genome was not static. It was selectively frozen, certain regions evolving at the slowest rate ever recorded in any vertebrate, while other regions changed normally. Not stopped, selective. Something was controlling which parts of this genome moved and which parts did not. The living fossil label had never asked that question. Now it had to. The 18th of April 2013, a paper landed in Nature, volume 496. Chris Amamiya at the University of Washington, alongside Jessica Alfeldi at the Broad Institute of MIT and Harvard, had done something nobody had fully managed before. They had sequenced the complete coelacanth genome. What they found was not what anyone expected. The protein-coding genes, the genes that build the animal, evolve at the slowest rate ever recorded in a vertebrate, 8 times slower than a human genome, 8 times. The coelacanth's body is not drifting. It is almost standing still, genetically. That is the comfortable part. But here is where the paper stopped being comfortable. The regulatory regions, the sections of genome that control when genes switch on and off, are not frozen. They are active, they have been changing. And buried inside them, Amamiya and Alfeldi found something specific, a sequence called the CSB enhancer. That sequence is not obscure. In tetrapods, in frogs, in mice, in humans, the CSB enhancer does one thing. It activates Hox genes, the genes that build limbs. The coelacanth has it, intact, functional, sitting inside an animal that has never grown a single limb. Remember what this record opened with, the 22nd of December 1938, a net hauled up from the Chalumnae River mouth. A fish with 4 lobed fins that moved like legs, the left front paired with the right rear, exactly as a 4-limbed animal walks. The question was never whether the coelacanth was connected to the origin of limbs. The question was how. The 2013 genome answered that, and then immediately made it stranger. The connection is not ancestral memory. The molecular machinery to build limbs is present right now, in living coelacanths, in the deep water off the Comoros. The switch exists. It has existed for 400 million years. It has never been pressed. What the index concludes, the coelacanth is not a creature evolution forgot. It is a creature carrying a door it has never opened, and biology cannot yet explain why.